So our next speaker is Dan Theodorescu. There's an enormous number of things I could say about Dan, but we'll start with the fact that he's the Cancer Center Director for the University of Colorado at Anschutz. He's done a bunch of interesting work. So he's identified a transcriptional signature for a GTPase called RalA. He's pushed work through actually to the point of clinical trials where it actually starts to see if the extent to which it matters in the clinic and actually drive translational improvements. He's been recognized in many ways. So he's an elected member of the American Society for Clinical... I can't read my own handwriting, and he was too modest to put it into his bio, so I'll let him do that one. I can read the American Surgical Association and also, importantly, the National Academy of Medicine. So with that, Dan, we look forward to hearing from you.
Great. Well, great. Well, thank you very much. Thank you very much, Larry, for inviting me. I feel like I'm the country doctor here, with all this august audience and fantastic speakers. But I'll try to make a good showing. My laboratory, I'm a urologist, and I also have a lab. And what we're interested in is really identifying the molecular mechanisms of cancer progression and translating those mechanisms into viable therapeutics. So I'm going to start with a little introduction. I don't think this slide is going to surprise anybody. We live in an incredible era of precision medicine. And what once used to be basically a stethoscope, and you see William Osler here say that medicine might as well be a science and not an art, and all the way down now with having the genome having been sequenced, we're in a massive transition, a tectonic shift in how we practice medicine. Most of us, however, are still here. We're still in the era of stratified medicine where we can actually separate people as groups as opposed to saying Mrs. Smith has this cancer and she has this molecular lesion and we're going to treat. But we're going there, we're getting there very, very, very quickly. So cancer is really a disease of the genome. And it's a disease of the genome really that is a compounding of genetic and epigenetic aberrations in somatic cells as opposed to germ cells. And the genetic aberrations that people talk about, and you've heard, of course, are really mutations, amplifications, et cetera. And both of these genetic changes and the epigenetic changes fundamentally conspire to change gene expression. Okay? And then this gene expression profile leads to, obviously, have an alteration in the proteome of the cell, the machinery of the cell, the metabolics of the cell, and ultimately that leads to loss of autoregulation, which leads to uncontrolled growth, invasion, and metastasis and spread, which are the bad things that cancer can do to you. So to put it in a cartoon form, what this is that this is a normal cell, and this normal cell is a well-behaved cell. So it doesn't grow unless it needs to. It doesn't move unless it needs to, say in a wound repair, for example. But ultimately, it's very well behaved, and it's basically well behaved because when it, for example, moves to repair a wound, when it hits the other side of the wound and the wound starts contracting, the cell doesn't move anymore and doesn't grow anymore. This autoregulation. And that autoregulation, these negative feedback loops, if you will, basically maintain this as a well-behaved cell. The problem is that in cancer, you have not a well-behaved cell, and that misbehaving cell is one that has lost these autoregulatory circuits that then allow it to do uncontrolled things like growth and metastasis, which means spread to other parts of the body. And ultimately, we're pretty good in medicine to destroy primary tumors. We've been pretty good at doing that probably since the 1950s and the 1960s when we really got surgery and radiation to really destroy primaries. What we're not as good at, and of course getting progressively better, is to actually get and kill, identify and kill metastatic disease. Because by definition, it's spread in many parts of the body. In some place, and sometimes we can't even see where it is with current technology. So in order to be able to do this, what we need to do is leverage the information of the genetic and epigenetic abnormalities of cancer cells, and then develop technologies such as small molecules, for example, that can then interrupt these pathways that, without autoregulation, have been left to basically their own devices and the cell in a state of uncontrolled growth or metastasis. So the idea is to develop molecules that attack various of these altered molecules, either at the mutation level or expression level, either genes that are overexpressed that shouldn't be expressed, or genes that are mutated that should not be mutated. So now I'm going to focus on my area of interest, which is really bladder cancer. Right. Okay. Yeah, I had her too. So apologize for that. I think my phone was I had a flood warning. Yeah. It was very close to the microphone. So my area of interest really is, as I said, to identify the genetics and the processes underlying tumor growth and metastasis. But because I'm a urologist and because I've had an interest in bladder cancer ever since I was a graduate student, I pursued that. And so I'm going to show you paradigmatically using bladder as a model. But you can basically extrapolate these kind of concepts to other cancers. So in bladder cancer, as I said a minute ago, not unlike other cancers, metastasis is really what kills people. And unfortunately, either with chemotherapy or even with immunotherapy, checkpoint inhibitors to be exact, we still have a lot to go. These are patients that don't do well. So we have further progress to be made. And the way that I envision progress is to really identify those targets that we can develop small molecules, either to alter the biology of the cancer or potentially enhance other therapies, such as chemotherapy and such as immunotherapy. And we're both working in both of those areas. And so targeting these genes would be the translational output of really understanding the biology. So we've used historically in our laboratory two approaches to identify such genes and then translate them to clinical biology and therapeutics. And one of them is basically to look at the cornucopia of genetic, epigenetic, transcriptional, metabolic data out there and distill and consolidate that data into accessible sort of packets that we can then probe at the functional genomic level. And we've identified several genes that way using functional genomic screens. Or the second approach is to develop animal models of tumor growth and metastasis that then we can query at the molecular level to inform us and then anchor that information from the animal models into human biology to get targetable agents. And today, I'm going to show you an example of using approach number 2. So in doing this, we used a bladder cancer cell model, several models, this is showing just one. We basically used models that were derivatized, if you will, from a non-aggressive, non-metastatic cell line through serial passaging in animals to derive very aggressive and metastatic cell lines. And then we did, in this cartoon, transcriptional evaluation of what are the differences in the genes that go up or go down as a function of this difference in metastatic capability across the cell lines. And when we do this, as you can imagine, there's two sides of a coin, genes that go up as a function of metastasis and genes that go down. And for today's discussion, I'm going to discuss these 412, start with discussing these 412 that go up with metastasis. We studied both sides, as you can imagine. And so the next thing, as I alluded to a minute ago, is to figure out which of these 412 are actually the most relevant in human biology. And the way we did that is basically anchor those 412 into basically querying which ones also go up as a function of aggressive disease in people. So we combined the list of genes that go up in the metastasis model with the genes that go up in the aggressive human tumors, and then did, if you will, conceptually, a Venn diagram. And we had 14 things left. And today I'm going to focus on RalA and RalB. So what are these? Well, it's very fitting that one of my heroes, since I was a grad student, is in the audience today, Dr. Weinberg, who identified RAS as the human. You better ask Dr. Weinberg that question. So he identified RAS, and RAS is upstream of RalA and RalB. This is obviously canonical pathways downstream of RAS. RAL is one of those canonical is one of those members, and the interesting thing is that the three major pathways, the MEK, RAF, and the PI3 kinase pathways, have small molecules against them, and therefore potentially having the ability to interrupt RAL is really useful as well. So we found RAL, and these are effectors of RAL. These are proteins that engage with RAL to go downstream and control a variety of cellular processes, such as cell growth, migration. All of these things, you can imagine how they could be related to metastasis and spread. So the reason why RalBP1, RAL binding protein 1, is actually in red is because it's the first binding protein that was identified for RAL, but also because of this. And this is a heat map analysis from a paper that was done by one of my collaborators. And what it is, it's one of the first papers that transcriptionally profiled a whole bunch of cancers, various histological types, right here. BL is bladder. And then looked at the genes in the array, and then did a cluster analysis to basically identify which genes are the most overexpressed as a function of tissue specificity. And you can see these wonderful red blocks. These are genes that are up specifically in that cancer. And without going into further detail, RalBP1 showed up in the bladder as a specific. So to make things as physiologic as possible, we basically looked at the binding partner that was the most relevant in bladder. So then we did functional analysis in bladder cancer, and that involved basically doing the classic things, which is pushing RAL levels down and boosting them with various molecular techniques. This is a Western blot, which quantitates the amount of protein, and we knocked down RAL and basically then looked at both in vitro growth in a dish or in vivo, which is growth in animals with various cell lines and various constructs. So in this case, for example, tumor size was in a mouse was basically pushed down, significantly reduced when you block RalA, and RalB had no effect. Similarly here, when we did forward experiments, we overexpressed RalA and RalB, you had some enhancement. And you could see that there are differences between RalA and RalB in some of these phenotypes, and I'm not going to go into that, but essentially, when you do these similar experiments for metastasis, and I show you the panel here for RalBP1, when you knock down RalBP1, the most relevant, if you will, effector of RAL in bladder cancer, you have a significant reduction in metastasis. So that gave a lot of credibility to the role of RAL in bladder. Of course, that's how we derived it. But what about other cancers? These are two studies, one from our group and one from a group at NIH, that looked at metastasis, and they looked at both RalA and RalB. And you could see here that if you look at our group, and this is in prostate cancer, metastasis is basically crushed when you reduce RalA, but not RalB. And a bone metastasis model showed similar results from the group at NIH. So it appears that RAL is not just important in bladder cancer, but in prostate as well. And this is data from Chris Counter, a colleague of mine at Duke, and he basically did animal models to show that lung metastasis is reduced when you knock down RalA, and even more so with RalB. So pancreas cancer seems to also be significantly regulated by RAL, which is not totally surprising because pancreatic cancer, 95% of those tumors have a KRAS mutation, And so, as you remember from the original, the signaling slide, RAS sits on top of RAL, so if you have a very active RAS, you have a likelihood of having activity in the RAL pathway. So lung cancer, this is a work from our lab, showed also an effect on relevance on the RAL expression. And so it would seem that both in human lines, such as this from our lab, as well as transgenic models that were basically published a few years ago from Chris Marshall, this is basically a model of lung cancer where you make transgenic mice that actually drive several cancer-causing genes in specifically lung cells. And these animals get lung cancer. One of these genes is KRAS. And so the idea then is these animals all get lung cancer because of RAS. Can you determine whether RAL is important? So what you do is you make knockout mice. So these are mice that are missing RAL at the full body missing RAL at the germline level, and then you cross them, and then you ask the question whether the progeny have the same incidence of lung cancer or reduced. And what this data shows is that you have a significant reduction in lung cancer when you knock out RAL. So again, very good information, very good evidence that coming from various directions, multiple tissue types, multiple models, human, mouse, etc., you basically have the relevance build. You're making a case for RAL being important experimentally. But what about human cancers? So this is a cartoon showing the GTPase cycle, and RAL being a GTPase, just like RAS. This is the cycle, so it goes from an inactive form to an active form that then binds the RAL-BP1, and that complex in that active form is the one that does all the damage and has the effect on the downstream signaling pathways. So in studying RAL, you have various ways to do this. You obviously can look at it at the DNA level for mutations, RNA for expression, protein for expression, and also activity of basically the genes that RAL is, if you will, inducing or repressing their transcription when it is active. So we wanted to do that in human tumors, and we started with an expression level in bladder cancer, and you can see this here. And we looked at the expression of RalA and RalB as a function of stage. And these are two studies, and this is a study from our own lab where we probed the levels of RAL as a function of tumor stage, and you can see that RalA goes up as a function of more aggressive and invasive bladder cancer. Interestingly, RalB did not change as a function of tumor stage, and perhaps even more importantly, there are only 3 RalA and 5 RalB mutations in 1,400 cases of bladder cancer. So clearly not a highly mutated gene, but one that is overexpressed. Work similar to this at the protein level and RNA level from the Michigan group revealed that RAL was relevant and upregulated as a function of aggressive and metastatic disease in prostate cancer. So this was similar to the animal experiments I showed you earlier from our group and from the NIH that showed that RAL was important in metastatic disease models. The cartoon lines up whether it's experimental or human. So for lung cancer, these are Kaplan-Meier survival curves. For protein, which is immunohistochemistry, and you can see for RalA and RalB here, and at the RNA level, that there are differences in outcome in lung cancer as a function of RAL expression, both at the RNA and protein level in lung cancer. So now the question is, how can we look at activity? And so activity is tricky, and there are a couple of ELISAs that have been perfected, but they're very difficult it's impossible to do them in formalin fixed material. So what I really wanted to do is establish the relevance of RAL activity in historical data sets, and there are many in the public domain. So I wanted to basically develop a gene expression signature of RAL activity, and the way we did that is we transcriptionally profiled cells where RAL was overexpressed or knocked down being shown here, and then transcriptional analysis done on those cells, and then we use the genes that separated neatly between RalA manipulation, RalB manipulation, or both, and looked at those genes on historical data sets that were in GEO and other public data sets. So when we did that, here's the data in bladder cancer, and it was quite interesting because the activity tracked also with tumor stage. In several sets, you see here, 4 different data sets, all are significant. And in some ways, the activity signatures tracked a lot better than the actual levels of RAL, both at the RNA and protein level. And that's not totally surprising because if you remember that GTPase cycle, most of the RAL the RAL that does damage is the active portion. And that portion is actually a very small component of the overall. And so RNA and protein measures the overall. The activity signature measures really the impact of the active moiety. So this is making sense. Similarly, we applied the signature on bladder cancer as a survival, and also in tumors that progress from non-invasive to invasive. And you can see here the tumors that progress look more like tumors that are already advanced compared to non-progressors. And this is survival after cystectomy or transurethral resection, which are 2 ways we can treat bladder cancer, either by scraping it or by removing the bladder. Prostate cancer, similarly, in advanced disease after removal of the prostate or invasion or non-invasion of the seminal vesicle, SVI, basically the signature was different between those groups. So clearly, it reported biologically and clinically relevant activity. So in summary then, in terms of the biological and clinical relevance of RAL, we can make the following conclusions. I think RAL is a driver in many human cancers, and also its expression activity does stratify patient outcome. So I think these are fairly solid assumptions. And given how many tumor types and how many samples were analyzed, I thought it was a fairly compelling data set to go on and try something fairly exotic that we had not tried before, but we wanted to basically discover and try to find small molecules that inhibit this GTPase. So this is the thing that I was faced with about 7 years ago, trying to figure out how to do this. And there's not a lot of clues of how to go about this approach. A number of groups had tried to drug RAS and tried various maneuvers to really go at the active form of RAS, which makes a lot of sense because RAS is very mutated. RAL is not mutated, and it wasn't really obvious to the field at that time how really to go after a non-mutated GTPase. So the idea was that I had one day, and this was driving home, and I had no real reason to think this could work, except there was a bit of a fantasy at that point. So the idea was that I hypothesized that the non-active form of RAL had a groove. That's a cartoon. This is the imagination part. So the cartoon here, and then when the RAL gets activated, that groove, in my mind, should actually close. And if I could only block that closure, I could then trap the RAL in the inactive form. So that assumes a lot of things. Number one, there is such a groove, that groove, if it does exist, closes with activation. So the fantasy was like this. Okay? So you basically have a small molecule, goes in that groove, and prevents this binding and this unpleasant cascade of downstream activators that then can cause all the damage by RAL. So put another way, those of you that are old enough to remember Pac-Man, basically this is the Pac-Man right here. These are the guys. And so what I wanted to do is basically this. Find something, you put a stick right here, and see all of a sudden these guys are smiling because they're not going to get eaten. Okay, so that was the idea in essence. But then this is where serendipity in science works out because those fantasies about RAL having a groove in the inactive form were actually true after I was able to convince the computational biologists to really look at the structures and see if they could find such a groove. And that was not trivial work, but they did. And not only was there a groove there, but this groove collapsed with activation. So it seems then that we had the perfect reagent, if you will, to test the hypothesis that if we dock a small molecule into that groove, it then can potentially hold the Pac-Man's mouth open. So we did this, and we screened this using a computer, and came up with 88 hits. So the question is, what do you do with those 88 hits? And well, there's a process when you do drug development characterization. And before I go into that, I'm going to show you this, which is the RalA and RalB are very similar in structure. So by all accounts, the molecule and the models I forgot to mention were done on RalA. The model, the likelihood that the molecule would block RalA and RalB was very, very high. And so we subsequently have structures, and we can say that the molecules bind in the same place. But when we did this originally, we did not. So what we went on to do is basically do a number of assays for the hits, and I'm going to show you some of these. But essentially, when you do drug development, you go from a very reductionist approach, where you have a molecule in a dish with just two proteins, to something more cellular, and then more animal, and then people, hopefully. And so I'm going to take you through some of these assays that we did. So the first assay was the alpha screen, which is really the biochemical, basically very reductionist. So this is basically a fluorescent assay. The only important thing on the slide is that if there's a molecule that actually gets in between and binds on RAL and blocks the interaction between RalA and RalB, there's a change in the fluorescence. And that's all it is. It's very reductionist. It doesn't mean it's going to get into a cell. It doesn't mean any of that. It means that it's able to break that interaction or prevent that interaction from happening. So once the 88 molecules passed that, a number passed that, and then we went on to do an ELISA, and an ELISA for RAL activation. So this is a bit of a more stringent test because what we used here are cell lines that have been engineered to be very good detectors of activated RAL. And I'm not going to go into the details of how this is done. Suffice it to say that this is basically assessing the binding of RAL to RAL-BP1 in a live cancer cell. So by virtue of the drug working there, that means, by definition, that the drug gets into the cell and blocks RAL activation. So we did that, and you could see an example of a hit here that suppresses RAL activation. Obviously, we use a cell line that has very high RAL levels as the sensor. So then I was talking to one of my very, very good friends and collaborators, Martin Schwartz, when we were both at UVA, and he had been working on RAL a fair amount at that time, and he had an idea when I was telling him about the various ways that we can test the molecules. He had a way to use caveolin knockout mice and wild-type murine embryo fibroblasts from caveolin knockout mice and wild-type mice because he had found that these cells were exquisitely sensitive to RAL manipulation. And so if they're sensitive to RAL manipulation at the molecular level, why not actually use them to screen the drugs? And so we did. And this is an example of RBC6, RBC8, and RBC10, where you could see that the assay here is that based on the caveolin status, that there's less spreading of the drugs from caveolin knockout that has no effect to the wild type, there's a difference. So we use this, which was kind of nice because, again, this was not a cancer, this was mouse. And then you're complementing all these assays. So it worked in the cancer cell to reduce RAL activation, and it worked in changing a downstream phenotype that is associated with RAL in a normal murine system. Okay, so to summarize then, we identified RBC6, 8, and 10 in the ELISA, and then this is the MEF spreading assay. So our three leading candidates were here, and then we decided to do some very important validation, because one of the things that we hadn't really seen, had done so far, is really figure out if the drugs actually bind to the spot that we had screened against using the computers. So it is conceivable that these would inhibit, some of these drugs would inhibit RAL, but they were binding somewhere else, not in the pocket that had been computationally found and we docked into. Fortunately, this did not happen. They bound in the pocket in the inactive form of the molecule. And the other thing that was really important is that this pocket and where they bound was actually different from the nucleotide binding pocket. And the reason why that's important is because the family of GTPases is large, 50 plus, 60 plus members, and many of them are very highly conserved in that spot. That's basically where the nucleotide activation occurs. So if the drugs had bound here, basically that could have been a problem, because you can imagine the toxicity if you inhibit 50 different GTPases potentially. So that was not the case, which was good. And number two, we confirmed the fact that we're binding in the spot we had screened against. And the results, and this was an NMR, were similar for the three leading candidates. So now the question arises, and this is a big one in drug discovery, is specificity. How specific? And that's a tough problem. When people do kinase inhibitors, for example, they use the kinase inhibitors and they have huge panels of kinases that they could see what their drug inhibits or not, and that gets at some of the specificities. But I'm not a pharmaceutical company, so I had to think of a much cheaper way to figure out if if this drug is specific or not to RAL. So we used multiple approaches, but I'll show you two. One is, I showed you earlier some slides that looked at lung cancer in human cells, and we screened a lot of human cells, and many of those cells were sensitive to RAL, but there are some that were very resistant. So, and that's with SI inhibition or SH inhibition and depletion of RAL. So that was molecular. What about the drug? So if the drug was specific, then the effect on those cell lines using the drug should mirror their sensitivity to RAL depletion using molecular techniques. And we picked 2 sensitive and 2 resistant lines from that paper, and we were pleased to see that the resistant ones were also resistant to the drug, and the sensitive ones were sensitive to the drug, both RBC8 and BQ57, which is a derivatized form that we made in the lab. So the other thing that we wanted to do is to actually put in an artificially mutated RAL, and the reason why that is good is because that active form will not have a pocket, which only occurs in the inactive form. So although that mutation doesn't occur in nature, it's a very useful tool to make sure that the drug is specific to RAL. And so the expectation would be that when you put this activated form and mutated form of RAL in cells and then hit them with the drug, they should be impervious to the drug because the drug's not going to knock out this artificial construct. And these are a little cut off, but essentially what it shows is when you put in this mutation, that the mutant line, the ones that have RalA G23V and RalB G23V, are basically insensitive to the drug, which was, again, a very good demonstration that the drug was specific. So then what we wanted to do in preparation for more significant animal studies was to figure out, we knew the drug was going into cells, cancer cells and normal cells, murine cells and human cells, but what we wanted to do is basically figure out if it gets into a tumor inside an animal. So we grew cell lines into tumors in mice and then removed the tumors as well as other organs and then carried out mass spectrometry that identifies the chemical structure of the compound in both RBC8 and several derivatives that now we've made. And as you can see here, the compounds go in various organs, but they definitely get into the tumor. So this is the sort of the beginning of pharmacodynamics, and the next is really the pharmacodynamic sort of testing, because now we know that the drug gets into the tumor as a bulk, but we want to know if it hits the target, because you can see the picture I'm trying to paint here is that eventually when we go to clinical trials, which I'm hoping we will, you're going to need to have all those tools available where you can probe the human cancer, not only to stratify it by expression, I'll show you all this at the end, but also whether it's inhibiting the target. So this is an activation assay by Western technology, standard activation assay that the field uses as a function of RAL drug delivery. And you can see here, there's a nice dose response for various inocula of the drug for BQ57 for both A and B, which suggests, again, that our ideas that the molecules are similar is consistent with that. So the small molecule inhibitors inhibit both A and B. And again, speaking to the issue of specificity, we had commercially available kits to look at activity of RAS and ROA to other unrelated GTPases, and these were not affected by the small molecules. So that was good, and now the question arose, what about xenograft experiments? So here's one example. We've done quite a bit more than this, but this is one example where we started a therapy, and you could see that the effect of the drug led to basically stabilization of the tumor growth in mice, and the magnitude of this is comparable to the level seen when you do depletion of the RAL using Si and shRNA. So we published this, and then we published several other papers looking at various derivatives, et cetera. But the good news is that companies can produce this drug and sell it without issues of patent infringement, and which is good because now the scientific community has been able to look at this compound in a variety of other models, and there are a multitude of papers now that have used these compounds to block RAL across various cancers, but also there's a paper that I'm author on that relates to glucose metabolism. And so that's close to being accepted, but that's another application in the field that was using compounds that were bought from a variety of commercial sources. One of the things I want to show you at the end here is that in addition to showing stabilization of tumors that are subcutaneous, we looked at this in a metastatic model, and this is really colonization, because for bladder cancer, we don't really have any models that do spontaneous metastasis from a human cell line. So we basically inject tumor cells in the tail vein, and then basically after various times, when we remove the lungs and do a DNA test for human DNA that we developed many years ago, we can see this curve of basically colonized cells that are growing in the lung. So the interesting thing was that when we actually started treating with these drugs, and we know from our work and the work of others that when you block RAL, it's even more impactful on colonization than actually it is on subcutaneous tumor growth. And interestingly enough, when you treat with the drug 2 weeks after inoculation, you have complete elimination of colonization. But the further out you leave it, the more the cells are able to colonize, which suggests that RAL is very important in the early steps of metastatic colonization. And you can imagine a scenario, I think it's the last slide now. Oh, no, you can imagine a scenario how you could use potentially that drug in the clinic. I'll show you that on the last slide. But before I show you that, the clinical application is that, in conclusion, I believe that RAL is important in cancer biology. And the concept that we showed here of basically trapping a GTPase in the inactive form using small molecule technology is something that could potentially be applicable to other GTPases that are not mutated. And there are many, many of those, and I have not looked at all of them, but I'm sure that somebody could look at the GTPase they're interested in and see that. And so we're very interested, obviously, in moving this forward. A patent has been issued recently on the original, and now we have patents in play on the derivatives. And so the fantasy clinical trial is here. So you take a patient that has had an extirpative operation, in this case, cystectomy, but you can imagine could be a Whipple operation for pancreas cancer, colon cancer, KRAS mutant. You look at the KRAS mutant status, for example, and then you look at the level of RAL by protein. It has to be high, and we've perfected the IHC for that. Then you look at the activation signature. You want that to be high. Then, because of it stratifies, as I showed you earlier, the outcome. Then, when you have a mutant RAS, you have high levels of RAL, and you have an active RAL. So you basically, what you're trying to do is, conceptually, you want to enrich your population in clinical trial patients. And the reason for that is very simple, because to quote, I don't know whoever made up this line, but in the pharma industry, you want to fail early and fail cheap. You don't want to mess around and do, and people have messed around, have done phase 3 clinical trials that have failed spectacularly, and those cost hundreds of millions of dollars. You want to fail early. So my vision is basically to highly enrich the population of whatever cancer we decide, or whoever has this drug decides, and then basically randomize them to either maintenance drug or standard of care. And if you stratify the patients well in patients that have very poor outcome, you're going to get an endpoint pretty quickly. And I think the adjuvant setting would be the best, because as you've seen from the metastasis data, that is a very, very compelling application for RAL. Conversely, we're also doing combinatorial functional genomics to find out what are the best combinations of RAL inhibition with various other agents, chemotherapy, immunotherapy, et cetera. So this is the idea of the trial. This is the fantasy made up Kaplan-Meier curve that I'm kind of hoping for. RBCA patients would be here, and the standard of care would be the outcome. So the most important slide is that, my dad was a math professor, and he could sit around with his graduate student postdocs and just write stuff. He was a theoretical guy, so he could just write papers around the table. I can't. I can think of stuff, but unless I have a team science approach to it and have people that I can convince, cajole, bribe, grovel occasionally, I can't do it. So this is the group that deserves all the credit. Thank you very much.
So we'll have time for one question. And while we're getting the question going, if you have a phone with you, which I suspect many of you do, if you could turn it to silent, that would be great, because the alarms are going to keep going off. Or turn it off if you're not sure how to silence the alarms.
So, pardon my naivete with this question. Hi, pardon my naivete with this question, and trying to draw a horizontal connection from this morning to this afternoon. So it's conceivable that in one of the somologic assays of blood or urine, they could detect RAL or some other protein even before cancer was clinically apparent or certainly not diagnosed, and then institute some kind of anti-RAL therapy or other kind of generic therapy so that cancer would never even be manifest. Is that theoretically possible or practically possible?
Yeah, I think detecting it would be definitely possible. I think the question is, if you detected it in a soma scan, the question is, what would it really mean? Because the heart of these GTPases is really the active component of them. And the active component varies to single-digit percentages to double-digit low percentages. So that's really the part that is the trickiest in order to be able to therapeutic target these. Because what you want to do, as I mentioned in the second last slide, is that you want to basically select patients that have a real clear evidence that the RAL axis and its activation is really there. You don't care as much that the overall levels of RAL, like for example, if you had an IHC that had relatively high levels, but then the activation signature was negative, I probably wouldn't put those patients on clinical trial because of the idea of fail early, fail cheap. You want to get patients that if you fail, and the drug, of course we're going to do pharmacodynamics, the drug gets in the tumor with biopsies. If it gets in the tumor and there's no response, and we do RAL activation assays, and we selected the patients well and it doesn't work, it's over. And I think that's the way we should do drug discovery in general. Oh, soapbox comment there.
Let's thank Dan.
Thank you.